Okay, so we're going to do the aerobic respiration lab. I'm going to read a little background information for you, and then I'm going to describe the materials and the procedure that you'll need for this lab. All active tissues are constantly using energy. Your body uses energy for walking, talking, eating, chewing, even moving your eyes. This energy comes from the oxidation of glucose. Oxidation is the process by which oxygen is combined with the substrate with the resulting release of chemical bond energy. In the chemical sense, oxidation is the loss of one or more electrons from an atom or molecule. Biological oxidation is a relatively slow process which can occur at reasonably low temperatures and occurs in a number of steps under the control of a specific enzyme. This controlled oxidation of glucose is called respiration. In respiration, a sugar is broken down into simpler molecules with the release of energy. This process may be represented by the following equation. Glucose plus oxygen reacts to form carbon dioxide, which is released, water, which is released, and energy. All right, human respiration and gas exchange. Carbon dioxide is a byproduct of respiration, and it's produced at the same rate that oxygen is being consumed. Therefore, we can use carbon dioxide production as a measure of respiration. In this experiment, you will measure carbon dioxide concentrations by the use of the indicator phenolphthalein, which changes color from pink in a basic environment to colorless in an acidic environment. Carbon dioxide, when mixed with water, will form carbonic acid. As the carbon dioxide concentration in water increases, so does the amount of carbon acid. All right, so for this lab, you're going to need distilled water, 1 straw, phenolphthalein, which is marked with a red P, sodium hydroxide, which is marked with a yellow sticker, NaOH, 2 flasks, okay, and a timer. You will have to share your sodium hydroxide and the phenolphthalein between 2 different groups, okay, because I just don't have enough to share. So for the procedure, you're going to pour 100 milliliters of distilled water into 1 flask. It doesn't have to be exact. It can be approximate. Okay, you're going to add 10 drops of phenolphthalein from the dropper bottle. If I can get it out. Four, 5, 6, 7, 8, 9, 10. It says if the solution turns pink, a very slight pink, you can assume that there is a little if any, carbon dioxide dissolved in the water. Okay, so there's no carbon dioxide dissolved in this water. You can place a white paper behind the flask just to make sure that it's clear. And it is. It's clear. If the solution is colorless, which in our case it is, we're going to add sodium hydroxide drop by drop to neutralize the carbonic acid until it has a light pink color. So we're going to add the sodium hydroxide until it becomes light pink. And it just took 1 drop. Okay? Once you obtain that light pink color, you're going to pour half of it into a second flask. Okay? One of these flasks will be your control. Okay, so we'll call the one on the right our control. Then what you will do, the other one is going to be your experimental. So what you'll do is you're going to sit quietly for 2 minutes. This is where your timer comes into play. So I'm going to just expedite this, and we'll say that I've been sitting for 2 minutes. You're going to put a straw into the experimental flask, and you're going to steadily blow bubbles into the water for 20 seconds. You'll notice that the solution loses its pink color. So do this for 20 seconds. Okay, so let's just say that's been about 20 seconds. Okay, it's clear again, the pink's gone away. Then what we're going to do is we're going to add the sodium hydroxide drop by drop, making sure that you keep track of the number of drops that you put in until we restore the pink color again. As you're dropping, you're going to stir. Okay, so it only took one drop that time. Then you're going to get your data sheet, which is this right here. You'll have in front of you. And on your data sheet, you're going to record the number of drops of sodium hydroxide that you put in at rest. So in this case, I put 1 drop. Okay? Oh, it went back to clear. So we're going to add 1 more drop. I think I'll put 1 more in there. There, our pink color is restored. So that was actually 3 drops of sodium hydroxide. Okay, now the next step, what you're going to do, and for this you're going to have to pick one partner to do the exercising and to do the blowing with the straw, okay, so that you just don't swap germs. The next step, you're going to exercise mildly by walking in place for 2 minutes. Don't be disruptive, okay? Try to pick a spot in the room somewhere, or maybe we can use the hallway, but just don't be loud and silly and disruptive. So what you're going to do is you're going to exercise mildly for 2 minutes, okay? And then you're going to repeat step 5 from above and record the number of drops. So after you've exercised, you're going to blow in your bottle again, or your flask, for 20 seconds until it turns clear. Okay, still, yep, we've got clear, okay. You'll blow for 20 seconds until you've got that clear color, okay? Then what you're going to do is you're going to restore that light pink color once again by adding sodium hydroxide. That was 1 drop. And that's 2 drops. By the way, when you're doing this, make sure that you have your phenolphthalein and your sodium hydroxide straight. Don't mix them up when you're adding the drops, or it won't work. Okay, I'm going to add 1 more drop. This is probably going to be the same amount for each. Anyway, so that now we have our pink color. It's going to be the same amount of drops probably for while I'm demoing this, because I'm not doing any exercising. Sorry. Alright, so now you've got your pink color restored again. Then what you're going to do is exercise vigorously for 2 minutes. This can include running in place, push-ups, sit-ups, jumping jacks. Jumping jacks are a good one. Okay, and again, don't be disruptive. You may use some of the space in the hallway, but don't be disruptive. You're going to do that for 2 minutes, and then you're going to come back and you're going to blow in your flask once again for 20 seconds, okay? So you'll blow in here for 20 seconds. Then you'll, once again, this will be clear. You're going to add drops of sodium hydroxide to restore that pink color, and making sure that you keep track of the number of drops of sodium hydroxide, and you'll record that under vigorous exercise. So you're going to be recording the number of sodium hydroxide drops 3 times. Once after rest, once after mild exercise, and then once after vigorous exercise. When you have finished gathering your data, you're going to make a bar graph with the data sheet that I've provided you. And then I'd like you to answer the following question on the lab. Okay? I think that's it.